Welcome back, everybody. It's hard to believe, but we've made it. We're at the end of our 90-day deep dive into the world of cellular receptors.
I know, right? From G protein coupled receptors to tyrosine kinases to nuclear receptors, it's amazing how much ground we've covered.
It's like we've been putting together this huge puzzle, piece by piece, and now we finally get to see the whole picture.
Exactly. And today we're taking a step back to see the bigger picture, like how all these different receptor families actually interact and what it means for disease and potential new treatments. It's a pretty tangled web, but we'll try to untangle it together.
Okay, so we always talk about receptors as if they're loners, right?
Yeah.
A signal binds, the receptor flips a switch, and boom, the cell responds. But I have a feeling it's not that simple.
Yeah, you're right. It's not that simple. One of the biggest shifts in receptor research has been moving away from this idea of like this global signaling, the idea that when a receptor is activated, the whole cell changes. Instead, we're realizing that signaling is much more specific, more like a control panel with very targeted effects.
So how did researchers even figure that out? It seems incredibly hard to observe something like that inside a tiny cell.
Well, research on cardiac cells, especially cardiomyocytes, gave us some really important clues. Scientists discovered that two different receptors on these cells, the beta-1 adrenergic receptor and the EP4 receptor, both activate the same signaling pathway, and it involves cyclic AMP.
Okay, same pathway. So you'd think they would both trigger the same response in the cell.
You'd think so, right? But that's not actually what happens. When you activate the beta-1 adrenergic receptor with adrenaline, the cardiomyocyte contracts harder. But activating the EP4 receptor, which also uses that cyclic AMP pathway, doesn't have that same effect on contractility.
So two receptors, same pathway, but different outcomes. That's definitely not a simple on-off situation.
Exactly. And this finding led to the idea that signals aren't just flooding the entire cell when a receptor is activated, but instead they're actually being directed to very specific regions or microdomains. Think of it like a city with neighborhoods, each responding to signals that are relevant to their specific function.
Okay, I can picture these tiny cellular neighborhoods, each with its own purpose. But how on earth does the cell keep everything so organized in there? It seems like things could get really messy really fast.
Well, that's where these things called A-kinase anchoring proteins, or AKE, come into play. They basically act as scaffolds within these microdomains, making sure that all the right signaling molecules are in the right locations.
So they're kind of like the city planners, making sure all the right signals get delivered to the right addresses.
Yeah, that's a great analogy. AKPs have a specific targeting domain that determines exactly where they are located in the cell, and they also have a binding domain that latches onto an enzyme called protein kinase A, which is super important in a lot of different signaling pathways.
It's just amazing how these tiny proteins can have such a huge impact on how cellular responses are organized. It makes you wonder how researchers even began to uncover these intricate interactions.
Well, it all comes down to understanding the fine details of protein structure. Scientists discovered that all AKPs have this common feature called an amphipathic helix. It's basically this coiled segment with one side that's attracted to water, what we call hydrophilic, and another side that repels water, what we call hydrophobic.
So it's like this helix with a split personality. I'm guessing that this sort of duality plays a role in how AKPs interact with other molecules?
You got it, exactly. It's that hydrophobic side of the helix that actually fits perfectly into a specific region on protein kinase. It's like this precise lock and key mechanism that allows AKPs to anchor that enzyme in specific locations.
That's a beautiful example of how the structure of something, even at the molecular level, completely dictates its function. This whole idea of microdomains and targeted signaling really changes the way we think about how cells communicate, doesn't it?
Absolutely. It challenges that old model of receptor signaling being this global event and highlights how important it is to understand all the intricate details of how these pathways work within these very specific compartments.
So we've talked about anchoring proteins in these microdomains in the context of heart cells. Are there other examples out there where we see this kind of localized control?
Definitely thyroid hormone receptors, or TRs, are another really fascinating example. There are two main types, TREA and TR, and they regulate a whole bunch of different processes in the body, from brain development to metabolism.
Thyroid hormones. They always seem like this kind of mysterious force controlling everything behind the scenes.
Right, and what's even more interesting is that they can act as both gene silencers and activators, depending on whether the thyroid hormone is actually present or not.
So they're kind of like a dimmer switch for gene expression, fine-tuning the cell's response based on the specific conditions.
Exactly. That ability to switch back and forth between silencing and activating gene expression is critical for maintaining that delicate balance in the cell, what we call cellular homeostasis.
But what happens when these intricate systems get messed up? What are the consequences when something goes wrong with the thyroid hormone receptors?
Well, one really striking example is this condition called resistance to thyroid hormone, or RTH. It's a genetic condition caused by mutations in the thyroid gene.
Oh, RTH. Doesn't that cause a lot of developmental problems?
Yeah, it does. These mutations actually prevent the receptor from binding to the thyroid hormone properly, and that leads to all sorts of issues like problems with mental development, hearing, bone growth, a whole cascade of problems.
Wow, that really underscores just how crucial these receptors are.
Hmm.
It is interesting, though, that mutations in TR have such a clear connection to disease, but we haven't found any human diseases that are linked to mutations in TRA.
That's a really great point. It suggests that TRA might play an even more fundamental role, maybe so essential that any disruption to its function is just not compatible with life.
Wow, that's kind of a sobering thought. It really emphasizes the delicate balance that these receptor systems are maintaining. And speaking of balance, I kind of want to switch gears a bit and talk about another really important aspect of cellular communication, inhibition.
Oh, yeah. Inhibition. The flip side of activation, just as important as turning pathways on, is having the right mechanisms to hit the brakes.
Right. And when we're talking about inhibition, there's this one family of receptors that really stands out, the glycine receptors. They're basically the main inhibitory neurotransmitters in the spinal cord.
Unlike GABA, which is the big player in the brain, glycine is the crucial inhibitory neurotransmitter down in the spinal cord. It makes sure that all that neural activity stays under control.
It's like the yin and yang of neural signaling, providing balance and precision.
That's a perfect way to put it. And glycine itself is incredibly simple. It's actually the smallest amino acid out there, but its function, anything but simple.
Simple structure, huge impact. It's a theme we see over and over in biology, isn't it?
Uh-huh.
So how do these glycine receptors work their magic in the spinal cord?
Okay, so picture these specialized neurons called inhibitory interneurons. They release glycine as their chemical messenger, and these interneurons connect to what are called alpha motor neurons, the ones that control our muscle contractions.
So basically we have these inhibitory interneurons putting the brakes on the alpha motor neurons, preventing them from getting overexcited.
Precisely. It's like a super intricate dance between excitation and inhibition. It makes sure all our movements are smooth and coordinated.
Okay, I'm following. So glycine is released, binds to its receptors on the alpha motor neuron, and that sets off a chain reaction that ultimately inhibits the neuron's activity.
Exactly. That's it in a nutshell. The key is maintaining that fine-tuned balance between excitation and inhibition. And when that balance is disrupted, that's when problems can arise.
Like with hyperexia or startle disease, where people
Yeah.
have those really exaggerated startle responses to even the tiniest stimuli.
You got it. That's
Yeah.
a classic example. It's often caused by mutations in the glycine receptors that mess up their ability to bind glycine or to open and close properly. So the brakes aren't working like they should.
It's fascinating, isn't it? How a tiny molecule like glycine can have such a profound impact on our ability to move, react, and just interact with the world. And speaking of interacting with the world, I'm curious about how our understanding of these receptor systems, especially those involved in inhibition, has led to new drugs or therapies.
Oh, there are definitely some interesting examples. One really powerful one is strychnine, which basically acts as a competitive antagonist at glycine receptors.
Wait, strychnine. Isn't that a deadly poison? I remember reading about it in those old mystery novels.
That's the one. Strychnine is incredibly toxic. It basically blocks glycine from binding to its receptors, which totally disables that braking system we talked about in the spinal cord.
So instead of inhibiting those alpha motor neurons, it's actually doing the opposite, unleashing them, which leads to uncontrolled muscle contractions.
Yeah.
No wonder it's so deadly.
Exactly. It's a stark reminder, I think, of just how powerful these receptors are and what can happen when their function is disrupted, even a little bit.
It's both fascinating and a little scary how a seemingly simple molecule like glycine can have such massive effects on our biology. But it also shows just how important it is to understand these systems, not just for the sake of scientific knowledge, but for developing new treatments and therapies too.
Couldn't agree more. As we unravel all the complexities of these receptor systems, we're not only gaining a deeper appreciation for how beautifully designed they are, but we're also opening up entirely new avenues for therapeutic interventions.
It's definitely an exciting time to be in the world of receptor research.
Mm-hmm.
And I, for one, can't wait to see what discoveries are just around the corner.
It really is. And it's amazing how much our thinking has changed about these receptor systems, you know? For the longest time, we were so focused on individual receptors and pathways, but now we're starting to really see how connected everything is.
Yeah, oh wait, we've zoomed out and realized it's not just a bunch of separate signals, but it's this giant network of communication. And that crosstalk between all the different receptor families, that seems to be where all the really interesting stuff is happening.
Exactly. It's not just about redundancy or backup systems either. It's about how cells fine-tune their responses, integrate signals from different sources, and create this really coordinated response.
It really makes you think about how disrupting these systems, even in a small way, can have all sorts of unintended consequences.
Absolutely. Think about a drug that targets one specific receptor. We used to think that it would only affect that one pathway, but now we realize that it could set off this chain reaction that affects the entire network.
So a drug that's meant to help with one condition could accidentally trigger a bunch of other problems.
Exactly. And that's why it's so important to understand not just the individual players, but the whole network and how it all works together. It's kind of like trying to predict the weather. You need to take all the factors into account and how they influence each other.
And the more we learn about these incredibly complex networks, the more opportunities we have to develop new and more targeted therapies. That could help us do things like modulate specific pathways or even enhance those natural braking mechanisms we talked about.
Precisely. And that kind of leads us to what I think is one of the most exciting areas in receptor research right now, personalized medicine.
Oh yeah, personalized medicine. The idea of tailoring treatments to each person's unique genetic makeup and their specific receptor profile. It's a powerful idea, but I imagine there are some pretty big challenges that come with it.
There are. And one of the biggest hurdles is just the sheer complexity of it all. We're talking about thousands of receptors interacting in countless ways, all influenced by things like genetics, lifestyle, environmental factors. It's a lot to take in.
It's like trying to map out the entire universe of cellular communication. Where do you even start?
Well, genomics is definitely one of the most promising avenues. By looking at a person's entire genome, we can pinpoint the variations in genes that code for different receptors. That can give us clues about how sensitive they might be to certain drugs or if they're predisposed to certain diseases.
So genomics is like getting a personalized blueprint of a person's receptor landscape. But it's just one piece of the puzzle, right?
Eh,
We also need to figure out how those receptors interact with each other and with all the other molecules inside the cell.
you got it. And that's where proteomics comes into play. That's the study of all the proteins in a cell or an organism. By understanding the proteome, we get this more dynamic view of how receptors function in real time, how they change, how they're modified, and how all their interactions change in response to different things.
It's like watching all these tiny molecular dances happening in the cell, seeing how those interactions shift and change based on different cues.
Exactly. And the more we learn about these dynamic interactions, the better we'll be able to design really targeted therapies that can actually modulate those interactions to achieve a therapeutic benefit.
It's incredible to think about all the possibilities. But it must be a daunting task. It's not just about mapping out all these networks, but also understanding what all those interactions actually mean in terms of how the cell functions.
It is a huge task, but the potential payoff is enormous. Just imagine being able to predict exactly how a person will respond to a particular drug based on their unique receptor profile, or being able to develop therapies that target only specific groups of cells, which would minimize side effects and really maximize effectiveness.
It would completely change medicine as we know it. But to get to that point, we need a massive collaborative effort. We're talking about bringing scientists from all different fields together, along with clinicians, patients, technology developers. Everyone needs to be involved.
Absolutely. And it's not just about the science either. It's about addressing all the ethical considerations, making sure everyone has equal access to these new technologies, and educating the public about just how complex and potentially powerful personalized medicine really is.
It's a huge undertaking, no doubt, but it's one that holds so much promise for improving human health and well-being. And as we wrap up our 90-day deep dive into this world of cellular receptors, I'm just struck by how much we've learned, but also how much more there is to discover.
It's truly amazing, isn't it? It's a field that is constantly changing, and new discoveries are being made all the time. And that's what makes it so exciting to be a part of.
And as we've seen, these discoveries aren't just about expanding our scientific knowledge. They're about understanding the fundamental workings of life, unlocking new ways to treat diseases, and ultimately, improving the human condition.
It's a powerful reminder of the impact that scientific research can have on our lives and the world around us. And going forward, it's crucial that we keep asking questions, challenge what we think we know, and embrace all the things we don't know yet.
And that's really what drives scientific exploration, right? That insatiable curiosity, that desire to understand the world around us, and that unwavering belief that knowledge is power.
Well said. And I have no doubt that as we continue to explore these amazing molecular machines, we'll uncover even more incredible things. It'll give us new insights into how complex life really is and pave the way for a future where medicine is truly personalized and tailored to each person's individual needs.
It's really incredible when you think about it. All those tiny processes happening inside our cells every second, all controlled by these little receptors.
It is, yeah. And it just shows how important it is to keep supporting research in this area. The more we know about how these systems work, the better chance we have of tackling some of the really big health problems out there.
It's not just about finding new drugs either. It's about understanding what causes diseases in the first place, finding new targets for therapies, and developing more personalized approaches that can actually make a difference for patients.
Exactly. We need a multi-pronged approach,
Yeah.
Combining all these new technologies like genomics and proteomics with basic research into how receptors are structured, how they function, and how they interact with each other.
It's like we need to look at the big picture and the tiny details at the same time to really understand how complex these systems are.
You got it. And it's not just about the tech either. It's about getting scientists, clinicians, and patients all working together, breaking down those walls and making sure everyone understands the challenges and the opportunities.
It's about bringing together the best minds and the most passionate people to really push this field forward.
And that's where our listeners come in. By staying informed, asking questions, and supporting scientific research, you're playing a huge role in helping us understand these amazing systems and creating a healthier future for everyone.
It's crazy to think we've covered so much ground in the past 90 days, and yet it feels like we've just scratched the surface of this incredible world of cellular receptors.
It's true. We've gone from the surface of the cell all the way to the nucleus, uncovering the secrets of signal transduction, crosstalk, and that delicate dance of cellular homeostasis.
We've explored how receptors contribute to both health and disease, the potential and the challenges of personalized medicine, and why ongoing research and collaboration are so crucial.
It's been a wild ride, and I hope our listeners have gained a deeper understanding of how elegantly designed and complex these molecular machines are. I mean, they really do orchestrate the entire symphony of life.
I know I have. And as we wrap up this grand finale, I want to leave our listeners with one final thought. The world of cellular receptors is this vast and dynamic landscape, full of mystery and wonder. So keep exploring, keep asking questions, and never stop learning. The discoveries that are waiting for us have the power to change our understanding of life itself and revolutionize how we approach human health.
Beautifully said. The future of receptor research is incredibly bright, and the journey of discovery has only just begun. So stay curious, stay engaged, and join us as we continue to dive deeper into this fascinating world of cellular receptors. Until next time, keep diving deep.